Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the humoral and the cell-mediated immunity. So humoral immunity is the one which involves B lymphocytes, whereas cell-mediated immunity involves T lymphocytes. So the whole story starts from the bone marrow. Both the B and the T lymphocytes are produced from the bone marrow. So the bone marrow is going to produce both B and T lymphocytes. Now, the point is that their pre-processing takes place at a different place. So the pre-processing of B lymphocytes is done in bone marrow, whereas the pre-processing of the T lymphocytes takes place in thymus. Now, what exactly pre-processing means? Now, pre-processing is a phenomena in which basically the self-reacting lymphocytes are destroyed. So one is that in pre-processing, the self-reacting lymphocytes are destroyed. And second one is that they are given a specific reactivity. For instance, there are different types of B lymphocytes and different clones of T lymphocytes. Each different clone will be reactive to a specific antigen. Okay, so all the different types of clones are given a specific reactivity. Okay, so all the different types of lymphocytes, for instance B lymphocytes, will not react with all the antigens. So they are very specific regarding it. So that specific reactivity will be defined during pre-processing. So this is one thing. And the other one is that in the pre-processing, the self-reacting lymphocytes are destroyed. For instance, we don't want any B lymphocyte or T lymphocyte in the body which has the ability to attack our own body cells. If some lymphocyte has the ability to attack our own body cells, that is known as the self-reacting lymphocytes. So such types of self-reacting lymphocytes are destroyed during the pre-processing. So the pre-processing of the B lymphocytes is done in bone marrow, whereas the pre-processing of the T lymphocytes is done in thymus. Okay, now, so after the pre-processing, basically these B lymphocytes and T lymphocytes both go to the lymph node. Okay, and these lymph nodes are basically located in the lymphoid organs like spleen, thymus, and bone marrow. And they can also be located in some major organs like respiratory tract and the GI tract. Okay, so the B lymphocytes after the pre-processing and the T lymphocytes after pre-processing goes to lymph node. Okay, now what will happen when the B lymphocyte in the lymph node and the T lymphocyte in the lymph node are exposed to antigen? Whenever they are exposed to antigen, the B lymphocytes will finally produce antibodies, whereas the T lymphocytes will finally produce activated T lymphocytes. Okay, the B lymphocytes will produce antibodies and T lymphocytes will be converted into activated T lymphocytes on exposure to antigen. Now these antibodies and activated T lymphocytes, what they are exactly going to do? They are basically going to attack the foreign invader. So how exactly the antibodies work? Antibodies work by two mechanisms, which we will discuss in details in the next few parts. So antibodies basically have two mechanisms of action. One is directly killing the foreign invaders, and the other one is killing by activation of complement system. So antibodies can attack the foreign invader in two ways. Number one, they can directly kill them, or they can kill them by activating complement system proteins. And whereas activated T lymphocytes kill the foreign invader by cytotoxic T cells. Okay, so this is the simple story of the humoral and the cell-mediated immunity. B lymphocytes and T lymphocytes both are produced from bone marrow. The pre-processing is done at different places. B lymphocytes pre-processing is done at bone marrow, and T lymphocytes pre-processing is done in the thymus. After the pre-processing, both goes to lymph nodes, and where on the lymph node, on exposure to the antigen, the B lymphocytes will produce antibodies, whereas the T lymphocytes will produce activated T lymphocytes. And then the antibodies will attack by directly killing or by killing, they will kill by activating the complement system. Whereas the activated T lymphocytes will kill the foreign invader by cytotoxic T cells. Now this whole process mechanism is explained in this diagram in the Guyton. So this is the B lymphocyte and this is the T lymphocytes. Both are basically produced in the bone marrow. So to start with, we have hematopoietic stem cells. Now hematopoietic stem cells will give rise to finally T lymphocytes and the B lymphocyte. Now hematopoietic stem cell will be converted into common lymphoid progenitor cell. So these are the two hematopoietic stem cells, this one and this one. They are converted into common lymphoid progenitor cells. Now this common lymphoid progenitor cell will be converted into T lymphocytes, and this common lymphoid progenitor cell will be converted into B lymphocyte. Okay, so hematopoietic stem cell is first converted into common lymphoid progenitor cell. These lymphoid progenitor cells will be further differentiated into B lymphocytes and T lymphocytes. But before the final formation of B lymphocytes and T lymphocytes, the pre-processing is done. So this common lymphoid progenitor cell, which is going to form the B cell, so here is the developing B cell. So this developing B cell will go pre-processing right here in the bone marrow from where it is being produced. Whereas this common lymphoid progenitor cell, which is going to form the T lymphocyte, its pre-processing will be done in the thymus. So this is thymus. As already told, the pre-processing of the B cells takes place in the bone marrow, whereas the pre-processing of the T cells takes place in the thymus. So here in the thymus, pre-processing of T cell is done. Here in the bone marrow, pre-processing of B cells is done. So after that, this mature T cell and the B cell, they will go to the lymph nodes. So here it's the B cell and here it's the T cell in the lymph node, and this is the lymph node in the peripheral lymphoid tissue. Now this T cell and B cell, when exposed to the antigen, will be converted into activated T lymphocytes and will form antibodies. So T cell will be converted to activated lymphocyte, whereas B cell will form antibodies. Okay, so this is the whole mechanism. So down here, B lymphocytes are present, which means this is the mechanism of humoral immunity, whereas this is the mechanism of cell-mediated immunity, because here are the T lymphocytes. Okay. Now, I have just converted this diagram of Guyton into a pretty detailed points, present all the things on one page, so it becomes like this. So, now this is the same diagram. Now, same hematopoietic stem cells converted into common lymphoid progenitor cells. So here in the bone marrow, the pre-processing of B cells is being done. Pre-processing, whereas here in the thymus, pre-processing of the T lymphocytes is being done. And what does pre-processing mean? So in pre-processing, there are two things, as already told, that the B lymphocytes and the T lymphocytes develop specific reactivity against one antigen. And the second thing is that self-antigen reacting lymphocytes are destroyed. So that's the pre-processing. So there are 2 points to remember regarding the pre-processing of B lymphocytes and T lymphocytes. One is that as we know that the pre-processing of the lymphocytes takes place in bone marrow. But the point to remember is that during the fetal life, from the start of the fetal life till the mid-fetal life, the pre-processing of the B cells does not take place in bone marrow. The pre-processing of the B cells is taking place in the liver. So the point to remember is that up till the mid-fetal life, the pre-processing of the B cells will take place in the liver, and after the mid-fetal life, the pre-processing will take place in the bone marrow. And one interesting point regarding the pre-processing of T lymphocytes in the thymus is that the pre-processing of T lymphocytes basically starts in the thymus before birth, and it will be continued till few weeks after the birth. So the pre-processing of the T lymphocytes does not take place for the whole duration of life. It will just take place few weeks after the birth. After that, the pre-processing of the T lymphocytes does not take place. all the clones of the T cell lymphocytes are already been made. So the pre-processing of the B lymphocytes takes place in bone marrow, whereas till the mid-fetal life, pre-processing of the B lymphocytes takes place in liver. And the pre-processing of the T lymphocytes starts just before the birth, and it will be continued few weeks after the birth. So the interesting point is, if some person has thymus removed surgically due to any reason, it means that person will keep on. There will be no effect will be on the pre-processing of the thymus, because it just takes place few weeks after the birth. But if during this after the baby is born, and within few weeks, if the thymus is removed, then it has devastating effect on the pre-processing of the T lymphocytes. Then the T lymphocytes cannot function well. Okay, so you have to remember these points. Okay, now after the pre-processing, basically different T cell clones, these 3 types of clones, just to explain, are made. And we have 3 different types of B cell clones. So basically, after the pre-processing, different B cells and T cells have different reactivity ingredients, different antigens. It means different types of clones of B cells and T cells will be produced. So for instance, this green one, red one, and this orange one are produced, and here also the green one, the blue purple one, and this red orange one are produced. Now, these different B cells have the ability to be reacted against to different types of antigens. For instance, now these different clones are present in the lymph node, and on exposure to the antigen, not all the clones of the B cells are being activated. Just one type of clone is being activated. For instance, this blue one is being activated. So this blue one will be activated, and the antibodies will be produced. And here, on exposure to the antigen, not all the different clones of the T cells will be activated. Only the specific type of the T cell clone will be activated, which has reactivity for that specific antigen, and that reactivity was assigned during the pre-processing process. Okay, so basically, so here the red ones are being activated, and here the blue ones are being activated. So this activated T lymphocyte, the specific the T cell clone which is being activated by antigen, that will produce the activated T lymphocytes, whereas this B lymphocyte will finally produce antibodies. So there are many steps between the B lymphocyte activation and the production of the antibodies. So these steps are basically, whenever the specific clone of the B lymphocyte is activated by antigen, on exposure to antigen, this B lymphocyte will first be converted into lymphoblast. This B lymphocyte will be converted into lymphoblast, which will be converted into plasmablast, and then this plasmablast will be converted into plasma cell, and this plasma cell is going to produce the antibodies. So basically, antibodies are not directly produced by B lymphocytes. They are produced by plasma cells. So B lymphocytes will be converted into plasma cells. And what are the steps how the B lymphocytes are converted into plasma cells? B lymphocyte will first be converted into lymphoblast, and lymphoblast will be converted into plasmablast, and plasmablast will form plasma cell, and this plasma cell will produce antibodies. Okay, now it's pretty interesting that we have a lot of different clones of T cells and a lot of different clones of B cells, which have specific reactivity to different types of antigens. Now, it means that we need a lot of different clones of T cells and B cells, because we have a lot of different types of antigens. So how are they produced? Now, how exactly are they produced? Now, the question is that does these all different types of clones have a specific gene for them? For instance, if there are 100 types of clones of T cells, are there 100 different types of genes for these T cells and hundreds of different types of genes for these different clones of B cells? So now discuss that. So the answer is, it's not regarding the different types of genes. So each clone doesn't have a different type of gene. Rather, each clone has a different type of combination of gene segments. Now, what exactly is a gene segment? For instance, these are the two genes, this yellow one and this red one, present on the DNA, and these genes are related to the production of different clones of T cells and B cells. Okay, now if I see this gene is basically divided into 3 gene segments. This yellow gene is divided into 3 gene segments, green, pink, and this blue one. Whereas this red gene is also further divided into these gene segments, for instance, purple, yellow, and green one. Okay, now what will happen that these gene segments from these 2 genes will interact with each other in the manner that, for instance, this pink segment from this yellow gene and this yellow segment from this red gene, these both will form a combination, and these both will interact with each other, and they will decide one single type of lymphocyte clone. Okay, not this gene is going to decide one single type of clone. Rather, it's one segment interacting with any other segment of the other gene, and a new combination is formed, and they will decide a different clone. Okay, now just imagine, for instance, if these genes are being distributed into these three segments. Now just imagine the number of combinations these 2 genes can make. Okay, so that's how different a relatively smaller number of genes can give rise to a lot more of combination of gene segments. So that's how they are giving rise to a lot different types of T cells and B cells clones. So this is how it's working. Okay, now what exactly is happening at the moment when this antigen comes in the body? So this antigen, now these B lymphocytes and the T lymphocytes are present in the lymph node. Now this antigen is roaming in the body. Now what will happen is that this foreign body antigen will be eaten up by some macrophage. Okay, for instance, some tissue macrophage has eaten this up, or some WBC, white blood cell neutrophil, has eaten this foreign body antigen. Now these macrophages will take this antigen to the B lymphocytes and the T lymphocytes. What I'm going to tell is that the B lymphocytes and the T lymphocytes are located in the lymphoid organs, whereas antigen is roaming in some tissue. Now, who will catch this antigen and will take this antigen to the B lymphocytes and the T lymphocytes? Okay, so the cells which will catch this antigen and will present this antigen to these T lymphocytes and the B lymphocytes are known as antigen presenting cell. So the most important antigen presenting cell is basically a macrophage, because the macrophage will eat this antigen up and will take this antigen to the B lymphocytes and the T lymphocytes, and will ask these B lymphocytes and the T lymphocytes to activate their specific clone against this antigen and produce the activated T lymphocytes and antibodies. Now, what exactly this macrophage is doing? This macrophage is doing something very interesting. Now, this macrophage will eat this antigen, and after eating this antigen, after phagocytosing this antigen, they will just use their most antigenic part, and bring that antigenic part to the surface of the antigen presenting cell. Okay, now after bringing their most antigenic part on their surface, these macrophages will take that whole antigen and specifically their most antigenic part on their own surface. They will take that to the T lymphocytes and the B lymphocytes. Now, what is going to happen on the B lymphocytes and the T lymphocytes? Now, on the B lymphocytes have antibodies present on their surface, whereas T lymphocytes have specific receptor proteins on their surface. Now, this macrophage is going to present this antigen to the antibody present on the B cell surface, and this macrophage is going to present this antigen to the surface receptor proteins present on the T cells. So that's how this antigen is being presented to this B lymphocyte and T lymphocyte by the antigen presenting cell, by the macrophage, which interacts with the antibodies on the surface of B cells and surface receptor proteins on the surface of T cells. Okay, so this is above this dotted black line above was the mechanism of cell-mediated immunity related to T lymphocytes, and below this dotted line is the humoral immunity related to the B lymphocytes. Okay, so this was regarding cell-mediated and humoral immunity. Just one interesting point regarding this, that initially this one single clone will be activated. This clone is then going to be differentiated into plasma cells, and then these plasma cells will convert it into these antibodies. This looks like that this is going to take a lot of time. So let's discuss that. For instance, this is a developing B cell in the bone marrow. This B cell is going to be converted into different clones of B cells, B1, B2, B3. Okay. And these clones have different antibodies on their surfaces. Now this antigen, this green color antigen is there. Now this antigen will activate just one single type of clone, either B1, B2 or B3. So this antigen is going to activate B2 type of clone. Now this B2 clone will start dividing, and a lot of these B2 type of B lymphocytes are produced. Now these B lymphocytes, which are all of the same clone, will produce a lot more of antibodies, and these antibodies will then attack the foreign invader. Okay, this is how it is going. Now, the same process here in the other flow chart, the B cell is converted into lymphoblast. Lymphoblast is converted into plasmablast, and plasmablast will be converted into the plasma cells. And these plasma cells will give rise to what? These green colored antibodies. Now, their timing and division and the rate of division, that's the interesting part and the last part of this lecture. Now, each plasmablast will be converted into a plasma cell in 10 hours. Okay? Plasmablast divides into plasma cell in 10 hours. Okay? So one plasmablast will be converted into 2 plasma cells in 10 hours or 10 hourly. Okay, and this division is going to take place 9 times. So, for instance, after 10 hours, we have 2 plasma cells. Then after further 10 hours or 20 hours, we have 4 plasma cells. After 30 hours, we have 8 plasma cells. After 40 hours, we'll have 16 plasma cells. Now, this division will take place for 9 times. It means around 90 hours. So, 90 hours is around a little less than 4 days. So, finally, how many plasma cells will be produced? 500 plasma cells will be produced in 4 days. If one plasmablast gets divided into 2 plasma cells every 10 hourly, then in 9 divisions, 500 plasma cells are produced. So it's like we have, after 10 hours, we have 2 plasma cells, then 4, then 8, then 16, then 32, then 64, then 128, and then 264 something, and then around 500 and 12, I think so. So these around 500 plasma cells are produced in 4 days by Guyton. Now each of these 500 plasma cell will produce 200 molecules of antibodies per second. Now just imagine 500 plasma cells producing 2000 molecules of antibodies per second. Just imagine where the number goes per second production of antibodies. And in a minute, in an hour, it is just unbelievable amount of numbers. Okay, so that's how the B lymphocytes are going to multiply into a lot more B lymphocytes, and those B lymphocytes, each of these B lymphocytes will further be matured into a plasma cell, and that plasma cell will just keep on producing a lot more antibodies, just like 2,000 molecules of antibodies per second. Okay, so this is it. This was humoral and cell-mediated immunity. See you in next part.